Okay, from yesterday, what might be one component of that cell theory? If we don't know, you feel free to look. Well, doesn't it say something about the cell theory above? And there's one. All living things are composed of cells. Okay, cells come from pre-existing cells. And yes, they're the basic units of structure and function. That's right. So, how about an organ system? Because both of you gave an example. Yeah, one of you did. I can't remember which. So, what might be an example of an organ system? If you can think of it yesterday, we can think of it today. Well, you pointed at him, but you're saying that.
Yeah, because he answered it yesterday, but I'm going to answer it.
You're going to answer it today.
Yeah.
Okay. So, what is a cardiovascular system? Don't say, man, don't tell us an organ system. What makes up a cardiovascular system?
A heart organ.
Then we just said, don't just say organs. So,
Yeah, the heart.
that's one. Yeah, the heart.
Yep.
What else? Nope, that would be a cardiopulmonary system. You're not entirely wrong. It is an organ system, just not the one that you said yesterday. Yeah, so it would be cardiovascular system. That is an organ system. Okay. So, moving forward then. This is where we left off. Yes. Oh, I got the screen locked. That's why. Okay. Again, here we just said what the cell theory was. It's these three components. Okay? Oh, no, I'm sorry. That's the history. Come on. Why is this not working? It's not forward. It's not going backwards. There we go. There we go. There's our cell theory we were talking about yesterday. Okay. So different types of microscopes. Okay. One thing about it, you don't have to see what this is because that's already written on your paper. So if you really want to see something at the microscopic level, you would be using one of these types of microscopes. And one of the things, we start short answer papers in this class, and helping yourself out when you're taking notes over these will certainly be a benefit for that. All right. And with this, of course, we wouldn't have anything this elaborate at this level. You'd have to go to either a funded organization or that of a college / university, or maybe the private sector to someone who does medical research. You might find something elaborate. So you can see we got a scanning electron microscope. There's going to be I think transmission is on there, and a SEM, TEM, and oh, tunneling. There we go. Okay. Did everyone have this information that wants it? Okay. So what could possibly a downfall be of a transmission electron microscope over a scanning one? What's an advantage to this here? What does it tell you? What type of not vision, but what type of can you observe with this microscope here? What type of picture does it show you? It's in what? It's in what? Three dimensions, okay? Which might be critical, telling you, okay, your different axes, your x-axis, then of course up and down is your y-axis. Where is your z-axis? Nope, that's your x. This here's x. Up and down is y, but where's z? Yeah, front to back is your z-axis. That's what allows you to look and visualize things in 3 dimensions. So if this is an advantage over a transmission electron microscope, which it is, what might it have in common with that? What does the tissue have to be in order to view it? What does it say? Yeah, it have to be dead tissue or non-living. I believe it says that. Oops. I don't. It's okay. So likes to skip ahead quite a bit. Okay. Okay, so the cell must be dead, okay, just like that of a scanning microscope. But again, what's the advantage of that scanning electron microscope? We just told you 2 minutes ago. It can be utilized to look at things in 3 dimensions. All right. So what can we see is an advantage of a tunneling microscope? Yeah, you can look at live organisms. That's right. Okay, does everyone have, because I think we start getting on to this, and the cell membrane I think is on here as well, that we want to get to today. So I don't know why you might know this, but you might. The simplest single-celled bacteria, the single-celled organisms that are the most basic are called what? I said it inadvertently. Say that again? Nope, not prokaryotic. Bacteria. Okay? Bacteria are very simple single-celled organisms. Okay? But what do you think causes more problems? Bacteria or viruses? But bacteria are living organisms. What's true about viruses? Yeah, those are non-living organisms. They're not even living. Okay? But they certainly cause a lot more problems for humans and animals than what bacteria do. Okay, one of the last things we want to talk about, and we're going to utilize the board here to discuss that a little deeper in depth, is what this plasma membrane is and why it's important. Excuse me. So how far in depth did you discuss cells? I mean, in elementary, can you name any structures that are involved or inside of a cell that you've learned from elementary or life science? We have seen one example up here. A nucleus is one. We have another example, it's up there right now. Yeah, a membrane, plasma membrane, cell membrane, either one of those is fine. Yeah, there's mitochondria in there, that's true. Can you think of anything else? What's the fluid portion of a cell? Anybody know? That's your cytoplasm. That's right. Now, the moment you go any further past that, it's probably something you never talked about in elementary, maybe in middle school. There's a structure called the endoplasmic reticulum. Ribosomes is another one. And then Golgi apparatus. Probably all vocabulary terms that you have on your handout as well. Okay? So what we want to look at, just from first observations, what's this? Okay, yeah, it's a coffee filter. So is this permeable? So and you can't answer that unless you know what permeability means. And permeability means, can anything pass through this filter? Yes. What can pass through here? Liquid, can you be a little more specific? Water. Because what goes inside this filter? Yeah, coffee grounds. So you put this in the coffee maker after it has the water, however strong you want to make your coffee, and you start putting your grounds in here, you turn the machine on, the water comes down from the top, goes through this filter. The coffee grounds do not go any further. Why not? Because it's impermeable to this membrane. In other words, it cannot pass through. Coffee grounds can't, but like you said earlier, what can? Water can pass through here. So that's what we're referring to when we're talking about permeability, when some substances can pass through and others cannot. Okay? So I need to see specifically what we had up here. Okay. For writing things down, I think we'll stop there. But when it comes to this phospholipid bilayer, okay? Of course, the simplest way to look at that is when you look at most motorcycles or pedal bikes. They've got one thing in common. Of course, well, they can't have a motor on there, electrical motor or something to that effect, but what do motorcycles and pedal bikes have in common?
2 wheels.
They've got 2 wheels, so you call that a what type of sickle? Bicycle, because it has 2 tires on it. So if we're talking about a phospholipid bilayer, it must have how many layers? It would have 2 layers to it, because that's what's going to lead to what we call permeability. Now, we'll look at this perhaps in your book. So if you draw this out, fine. If you don't, that's fine too, because you can always look at this again in your book. Okay? So in general, and this would go all the way around. And again, that would go all the way around. So does anyone ever heard of these terms? Nope, that don't look right. Let me double check that. I was right the first time, darn it. We'll just leave it at those. There's actually 4. Has anyone ever seen those before? Diffusion, facilitated diffusion, and osmosis. Yes? Yes, sir. Has anyone ever seen these before? Okay. One of the things that I think maybe, there's a couple things. This one might not make the most sense. This one I think we can visualize, and then this one as well. But, for instance, for those of you who may, on CBS on Wednesday nights, so that would allow you to watch this show called Survivor. Does anybody follow that show? Okay, well, regardless then, we'll just use this as an example. If you are on the coastline, whether it's Florida, California, Hawaii, anywhere where you can get into the ocean. Anyone been in the ocean? So for those of you who have raised your hand and you've actually had be able to experience being and swallowed it. Has that happened? Yeah. Okay, so for those of you who have been in the water and have swallowed that, what do we taste? Salt, because the oceans on this planet are salt water. Okay, and if you take zoology, we talk about freshwater organisms and salt waters. Crocodiles is an example. You have saltwater crocodiles, and you have freshwater crocodiles. Tells you where you're going to find them. If you are a freshwater crocodile, chances are you're probably in Egypt and along the banks of the river. Or you could be in Australia. Those could be freshwater crocodiles as well. But our point behind that is if you are on an island, or I don't know what your history is with World War II, but I think it was called the USS Indianapolis. During World War II, that was a ship that was torpedoed by the Japanese, I believe. Okay? So you have all the crew that could survive that initial blast that are actually in the ocean, the Indian Ocean, I think, on the USS Indianapolis. Some of them survived for, I don't know, maybe 10 days. But of the crew that went in, let's just say 900 went in. Okay? Maybe there's an initial crew of 1,000. 900 went in the water living, and 360 survived. Okay? It could be for a multitude of reasons. What might be one reason that some sailors did not make it on the crew of the Indianapolis? Whether it's after the initial bombing or torpedoing of the Japanese, what might have caused some of those sailors to perish? No fresh water. Okay, so one might be from predation from sharks, but also no fresh water drink. Because if you're in the ocean water, we already established it tastes salty. Okay? So when we're talking about that, osmosis is a movement of water either into or out of your cells. So what it's going to do is follow the solute concentration. Okay? So in an individual's body cells, you have all this fluid in here. Okay? So again, you would have a nucleus in here, and then you have this. Okay? So what did we just verbally said earlier? What is the cytoplasm? It's a type of fluid found in each type of cell. But if an individual is in seawater and they continually consume this salt water, what happens to the concentration of the salt inside their body? Like if you had 6 cans of Dr. Pepper, 6 cans of Mountain Dew, you keep drinking those cans or bottles or whatever, the sugar concentration and the caffeine concentration, what does that continually do? Is your sugar level going to go down? No, because you keep consuming that soft drink or pop. So that's going to continually go up. So for the same type of logic, what is the amount of salt going to happen? What's that concentration going to do inside the body? That's also going up. So what we see happening here, people may inadvertently keep taking in this salt water or sodium chloride. So this concentration, as you said, keeps going up. Okay? So the fluid then that's inside the cell is going to want to balance what's on the outside. Okay? So if we have low salt in here, so low salt. Here we've got an increased amount of salt. We can just put higher here. Maybe that makes more sense. So we've got a higher concentration of salt on the outside, a lower concentration of salt on the inside. Okay? This isn't a movement of the salt, it's a movement of the water. Okay? So what's going to happen then to balance this out, okay, the salt's going to come in and water is going to continually be pushed out. H2O goes out and people will perish from this because the body can only take so much salt water in their body. And it's a specific term called that, but they just get so dehydrated that they can start hallucinating, and their nervous system will start to shut down. And it's of nobody's fault, it's just something that unfortunately can happen. So this, again, osmosis is the movement of water across a concentration gradient. Okay? So what we are trying to draw then, I see a lot of you have, yes sir.
Can I fill this up?
That's probably his bottle.
It is, but I don't want to go again. I don't want to ask to go to the bathroom.
Well, why didn't you do that the first time? Go fill that up. Okay?
It is right here.
All right. So those of you who do have your book, since you have those, let's go ahead and look at page 188 if you have them. Otherwise, if they're back there, go ahead and go grab them. Actually, page 187. So we're talking about the plasma membrane on page 187. Okay? Function of the plasma membrane. Recall that the process of maintaining balance in an organism's internal environment is called homeostasis. Please stop that. Thank you. Homeostasis is essential to the survival of a cell. One of these structures that is primarily responsible for homeostasis is the plasma membrane. The plasma membrane is a thin, flexible boundary between a cell and its environment that allows nutrients into the cell, that allows waste and other products to leave the cell. All prokaryotic cells and eukaryotic cells have a plasma membrane to separate them from the watery environments in which they exist. A key property of the plasma membrane is selective permeability, like we were talking about here with our coffee filter. Okay? By which a membrane allows some substances to pass through while keeping others out. Consider a fish net. It is an analogy of selective permeability. The net shown in figure 8, there's that 5. Figure 5 has holes that allow water and other substances in the water to pass through, but not the fish. Depending upon the size of the holes in the net, some kinds of fish might pass through while others are caught. The diagram in figure 5 illustrates selective permeability of the plasma membrane. The arrows show that substances enter and leave the cell through the plasma membrane. Okay. Control of how and when, how much of these substances enter and leave the cell relies on the structure of the plasma membrane. So what you see there in figure 6 is what we're trying to illustrate up here. I mean, it's not an exact replica, but hopefully we can see that of what the idea is. Most of the molecules in the plasma membrane are lipids. That's why we call it a phospholipid bilayer. Okay? Lipids are larger molecules that are composed of glycerol and 3 fatty acids. If a phosphate group replaces a fatty acid, a phospholipid forms. Okay? A phospholipid is a molecule that has a glycerol backbone, 2 fatty acid chains, and a phosphate-containing group. The plasma membrane is composed of a phospholipid bilayer. Okay? We're not done yet, people. Okay? In the plasma membrane, phospholipids arranged excuse me, in a way that allows the plasma membrane to exist in a watery environment. Okay? There's other aspects that we see that I thought would be to your advantage to see that in figure 6. Because if I draw this on here, you're going to have no idea what that means. But that is a transport channel? Transport protein. Okay? Because some substances that enter and leave the cell are just too big to enter and leave by diffusion alone. Okay? Sugar, carbohydrates, those are too large to enter and leave the cell. Gases like oxygen, carbon dioxide, and water, they can pass through very easily because they are such smaller in nature. The molecules are much smaller in nature. Okay? So, again, a phospholipid bilayer, you've got 2 sides to it. Okay? You have, it doesn't say, oh, it does say polar heads. Okay? That's what these are on the outside. These are polar heads. These are non-polar on the inside. Okay? Okay. I believe we will stop there for today. We will pick up with this tomorrow. So we will catch up to you next time.